want to especially welcome us all to today's clinical presentation. Before we continue, I would like to call on Pharm Dr. Bello to take us on the opening prayer.
Okay, can we pray, please? As we are about to start this meeting, we pray to God Almighty to come and be with us, to guide us, and to bless our deliberation. We trust in Him. Fatiha. Amen.
Amen. Thank you very much, sir. I want to first of all welcome on behalf of our acting HOD, Pharm Charles Nwakile. I want to specially welcome us all to today's clinical meeting. It's going to be a very educative one. I want to recognize everybody present here, all our distinguished pharmacists from different walks of practice. We'll especially welcome you and thank you for joining us today. I want to recognize the fellows of Pharmaceutical Society of Nigeria present here. I want to recognize HODs. I want to recognize the deans of different faculties, especially of the University of Ilorin. I want to recognize our PSN chairman present here. Thank you for joining us. And of course, all of us that are here today, thanks for joining us. We have a very, very important topic that we are going to be discussing today, and we have a seasoned lecturer to take us on this topic. And be rest assured, he's going to expand enormously on it. So I would like to introduce the speaker, and I'll be taking us on his citation. Our speaker for today is Toyin Abdullahi, and the topic for today is comparing COVID-19 vaccines. I will take us on a brief of the speaker for today. So, Pharm Toyin Abdullahi, B.Pharm, MSc, PhD, is a senior lecturer and acting head of the Department of Pharmaceutical and Medicinal Chemistry, Faculty of Pharmaceutical Sciences, University of Ilorin, Kwara State. Dr. Toyin Abdullahi is a senior lecturer and the current chair of Nigeria Association of Pharmacists in Academia, University of Ilorin Chapter. He completed his PhD in pharmaceutical chemistry at Obafemi Awolowo University, Ile-Ife, in 2017, and was appointed acting head of department in October 2018. He obtained his Bachelor of Pharmacy degree from Ahmadu Bello University, Zaria in 2001, before embarking on his internship training with Ahmadu Bello University Teaching Hospital Complex, Katsina State, from 2001 to 2002. He served as a corporate pharmacist with General Hospital Gaidam, Yobe State, from 2002 to 2003. Dr. S.T. Abdullahi worked with Worldwide Commercial Ventures Limited, a pharma logistics company now known as Worldwide Healthcare, from 2004 to 2012, before resigning to join University of Ilorin in July 2012 as lecturer 2, having obtained his MSc in pharmaceutical chemistry from Abu Zaria in 2010. His main areas of research interest include pharmaceutical analysis, clinical pharmacology, pharmacokinetics, pharmacogenetics, and pharmacometrics. Dr. S.T. Abdullahi is a member of the Statistics and Pharmacometrics Interest Group, West African Society of Pharmacology, Royal Society of Chemistry, Nigeria Association of Pharmacists in Academia, and Pharmaceutical Society of Nigeria, with publications in reputable peer-reviewed local and international journals. Please, I would like us to welcome Dr. Saad Abdullahi as he expands to us today on COVID-19 vaccines. Thank you, sir. You have the floor.
Thank you very much, Pharmacist Ngozi. A very good morning to you all, the acting head of pharmacy department, University of Ilorin Teaching Hospital, all other protocol duly observed. You are all welcome to today's update lecture, the title of which is Comparing COVID-19 Vaccine. As you all know, COVID-19 is one of the things that is currently trending all over the world. And we are going to look at what are the advancements that have been made, especially in the area of the vaccines. So the overview of my presentation is we are going to briefly look at how science has helped us to fight COVID-19. And then we look at what are the methods or the options that are available in the production of vaccine, the different types of vaccine. And then we now look at the current types of COVID-19 vaccine particularly. We'll look at how these vaccines work, what are the similarities and differences between them. Back in January 2020, Chinese researchers shared the SARS-CoV-2 genetic sequence for the virus that has since ravaged the whole world. And the genetic sequence was found to be approximately 30,000 nucleotide long. Back then in January 2010, we did not know that this pandemic will get to this magnitude. But right then, at that time, the scientific tools that we need to fight this pandemic has already been made available. The sharing of this genetic sequence has enabled us, it has helped a lot in the following area. First of all, in the areas of testing, because the PCR and the antigen test that is used to test samples rely on identifying the genetic sequence of this virus in the samples. And then now, currently, as we have seen, it has also helped in the area of vaccine design, because most of the vaccines that we will get to be seeing now rely on part of this genetic sequence that has been shared, especially as we have seen with the Pfizer and BioNTech vaccine, and also with the Moderna vaccine. And then it has also helped us now with the issue of monitoring mutations in this vaccine. Knowing the genetic sequence of this vaccine allows us to be able to track changes to the genetic sequence of this vaccine, so that as mutation occur, we know how this will affect the virus. As we all know, genes are made up of nucleotide. The genetic sequence of virus, just like the genetic sequence of human being that is made up of DNA, that of virus is specifically made up of RNA, and they are made up of 4 nucleotides: the adenosine, guanosine, cytidine, and uridine. This nucleotide they make up the genetic instruction for the gene. Just as we have the English alphabet that makes up words and sentences, nucleotide also make up the genetic instruction for the human being or the virus. The year 2020, it has shown a dramatic, extraordinary global effort in the development of vaccine, because currently we have greater than 60 vaccines that are in clinical development and more than 170 in preclinical clinical development, which is good news to the COVID-19. There are basically three main approaches to the design of vaccine, and the differences lie in whether we are using the whole virus or we are giving part of the virus that will trigger immune response, or we are using just a part of the genetic material that we provide instruction for the synthesis of specific protein that will trigger these immune responses. The whole microbe approach can involve use of inactivated vaccines, such as we have with the flu and polio vaccine, or we are using live attenuated vaccine, as we have in the measles, mumps, and rubella vaccine, chickenpox, or shingles vaccines, or with the vector vaccine that involves use of a vector and then that has been genetically altered, and then you can inject the genetic code of interest into the vector vaccine. Example of that is the Ebola vaccine that was approved in July 2020. The subunit vaccine involve using part of the genetics, part of the virus that can trigger an immune response. As we all know, the main essence of vaccination, the main principle behind vaccination is to mimic infection, but in a controlled way, in such a way that it can trigger immune response without causing an illness. And that will depend on the vaccine presenting a recognizable part that can trigger this immune response. So this part that can trigger immune response can be given alone. Example is the protein that will help the virus to invade their host and trigger immune response, or it can also be in form of sugar. Example of these vaccines are the whooping cough vaccine or the tetanus, diphtheria, and meningitis vaccine. And then the current trend now, which is the genetic approach that utilizes the genetic material of the virus or part of the genetic material of the virus in form of DNA or RNA. Here are examples of some of the COVID-19 vaccines that have been recently granted emergency use approval. The Pfizer-BioNTech vaccine and the Moderna vaccine are example of the RNA vaccine, whereas the Oxford AstraZeneca and the Sputnik V vaccine are examples of the DNA vaccine. I will now take us through what are these RNA vaccines and how do they work. The RNA vaccine, just as we have seen, contain the spike protein, which is the recognizable part that can serve as an antigen that, if injected, it can trigger an immune response. So we can take part of the genetic material in form of mRNA, in form of RNA. When a gene wants to perform its work, it produces its own a part of the gene in form of messenger RNA that can now be used to provide it, that can be used as an instruction to provide the work that the gene is being made to do. So the RNA, messenger RNA, can be synthesized. When we have our RNA molecule, the RNA molecule have the instruction. It is encoding the viral spike protein. So this messenger RNA, when given, can now produce the spike protein in our own body cells, which the immune system can recognize and then trigger the response. But the main problem with that is that how do we get the RNA into the body cell? The RNA, as we know, is very hydrophilic. And for it to cross the cell membrane, we need to tiny fat droplets. So we have the lipid nanoparticle that is encapsulating it, and then the lipid nanoparticle can also be further coated with polyethylene glycol, which can provide more stability to the RNA, the messenger RNA or the RNA as the case may be. So with the RNA instruction embedded in the lipid nanoparticle, we have a vaccine that is ready to be injected. So when this vaccine is now injected, it can get into the cytoplasm. And once it crosses the cell membrane and is in the cytoplasm, it can now synthesize the viral protein, the viral spike protein. The ribosome, which is the protein manufacturing unit of the body cell, is located in the cytoplasm. So the RNA does not need to get into the nucleus. Once it is being ferried into the cytoplasm, it can carry out the instruction and synthesize the viral protein, which can now trigger the immune response. So what are the advantages and benefits of this approach? The main benefit of this RNA approach to vaccine is with the pace at which these vaccines can be manufactured. They are very easy to synthesize in the lab, and once they are synthesized and encapsulated, you have your vaccine. So this pace is one of the main advantage. Take, for example, Moderna. They were able to design their own messenger RNA vaccine within 2 days after the Chinese scientists shared the genetic sequence. And after 25 days after they shared the genetic sequence, they have produced the first clinical vaccine for use. So the pace at which it can be produced is one of the main benefits. And then another benefit is the safety aspect. RNA on its own cannot cause infection. They are just instruction for the body, for our own body cells to synthesize a recognizable antigen. So they are quite safe. When given, they don't have to go into the nucleus. So it's not going to cause any mutation. They are just, once they get into the body cells, it can carry out the instruction. So that is why RNA vaccines were able to make it to the forefront, and they happen to be one of the first vaccines that were produced. Then the main challenge with this, as we have seen, is with the issue of storage and transportation, because RNA are very fragile, they are less stable, so they have to be stored at an extremely low temperature. So we have examples of this messenger RNA vaccine. We have two basic examples of the vaccine. The RNA vaccine, we have the messenger RNA vaccines, as we have seen with the Moderna, Pfizer, BioNTech, and CureVac vaccine from Germany. And then we have another type, which is the self-amplifying RNA vaccines from Imperial College, and another one from Arturus in Singapore. The main difference between the two is the presence of a viral enzyme replicase in the self-amplifying RNA vaccines. This replicase enzyme is able to make more copies of this RNA once they get into the cytoplasm. So you can give very little dose, very small dose of the vaccine that can trigger an antigen expression that can last up to 2 to 3 weeks, as against 2 to 2 days if it is the ordinary mRNA vaccines. So we have example of Arcturus vaccine from Singapore. Is based on this self-amplifying RNA vaccine, and because it can be given in lower dose, it is the Arcturus Therapeutics people are working on it to be a single dose vaccine. So these are the basic profiles of this RNA vaccine, how they work, and examples of this RNA vaccine. The second basic category of this genetic approach to vaccine are the viral vectors vaccine. The vector vaccine are similar to the RNA vaccine in that we are using genetic approach to it, but in this form, in the form of the DNA, we have the part of the RNA genetic material of the virus that is responsible for the spike viral protein production, and that part can be given in form of DNA. And then we need, instead of a lipid nanoparticle, we need a viral vector to serve as the vehicle instead of lipid nanoparticle, as we have seen with the RNA vaccine. The viral vector vaccine is meant to take care of the challenges that we have encountered with the RNA vaccine. That is the issue of the vehicle. We now use a viral vector. A viral vector is a vector that has been genetically altered. The gene that is responsible for its replication has been removed, so it is very safe, and then we can input the DNA into the viral vector, and then we have our vaccine. And then once we have that vaccine and it is injected, it can get into the cytoplasm. And then once it is inside the cytoplasm, the thing with the DNA is that they have to first of all get into the nucleus, and then the DNA can now produce it for itself in form of messenger RNA that can carry out the instruction of producing the viral protein that will trigger the necessary immune response. So what are the benefits of this vector vaccine approach? One of the benefits, as we have seen with the RNA vaccine, is that the pace, it is relatively easy because the DNA too can be synthesized. So the approach is faster compared to the traditional method of developing vaccine. And then the safety also, it is also relatively safe because the virus, the vector proteins, once it has get into the cytoplasm and the DNA has carried out the instruction, the vector particle is destroyed, it is broken down by the normal body process, so it is also relatively safe. The only challenge to this approach is the minor side effect that we'll get to see, because most of these viral vector, we have also been exposed to them, and then once we have been exposed to them, once they are now being given in form of a vaccine, the body can also trigger an immune response to it, and then that will result in form of minor side effects such as headache and fever. Then we also have two different types of vector vaccines. We have the replicating vector vaccines and the non-replicating vector vaccine. The main difference between the two is that The replicating vaccine will produce multiple copies of the viral vector particle once it is inside the cytoplasm. So we can give it at a lower dose, but the non-replicating will not produce more copies of the viral particle. So most of the COVID-19 vaccines are non-replicating viral vector vaccine so that they can be much more safer. So we have the different examples of these vector vaccines that are currently in phase 3 of the vaccine development. We have the Gamaleya Research Institute in Russia's Sputnik V vaccine that make use of 2 vectors. We have the human adenovirus vaccine, and we have different subtypes of this adenovirus vaccine. So the Russian vaccine makes use of 2 vector vaccine for the prime and the booster dose immunization. The Russian scientists were able to identify that some of these vector vaccine will also trigger an immune response, just as we have said. So when you give the prime dose, by the time you will be giving the booster dose, the body immunity will also affect the efficacy of the second dose vaccine. So because of that, they decided to design their own Sputnik V vaccine with 2 vectors, the type 26 for the prime immunization and type 5 for the second immunization. We also have the Johnson & Johnson from USA that has recently also been approved, has been authorized for use using type 6, and is a single dose vaccine. Then we have Cancino Biologics from China, which is also using type 5. And then the Oxford AstraZeneca vaccine decided to use a much more vector that is not common, using primate adenovirus vector, like the chimpanzee adenovirus. And then we have another vaccine from Italy, Retera, that also uses gorilla adenovirus vector. So those are the 2 main genetic approach to vaccine development, how they work, and the similarity and differences between them. Then we have examples of vaccine, COVID-19 vaccine that uses the traditional method. We have the inactivated vaccine from Sinopharm, and another one from Sinovac. They all work based on use of kill SARS-CoV-2 virus. They are inactivated, they cannot cause harm, but the efficacy, as we will get to see, is not up to what we have seen from the genetically engineered vaccine. So these are the 9 examples of COVID-19 vaccines that are almost, or that are in phase 3 of clinical trial. We have Pfizer, Moderna, with efficacy of approximately 95%. They are also to be given in 2 doses. And then we have the storage temperature of -70 and 20. The storage temperature of Moderna is relatively lower, -20. It is possible to modify some of these RNA vaccine during the design. Like, for example, the nucleotide sequence of Moderna vaccine is about 3,819 nucleotide long, compared to the Pfizer-BioNTech vaccine that is 4,220 something nucleotide long. So in the design of the vaccine can affect the storage temperature. Another thing that can be responsible for the difference in the storage temperature between these 2 RNA vaccines may be in the area of modification. We can modify the RNA by, for example, simple latching or inclusion of methyl group on any of the 4 nucleotide base type can modify the RNA vaccine and can affect the storage temperature. Then we can also improve the stability of this RNA vaccine by adding stabilizers and removing water in form of freeze drying, lyophilization. That can also improve the stability of this RNA vaccine. So it is possible that Moderna has used one or any of these 3 approaches to improve the storage temperature of their own vaccine. It's a proprietary right of the company. It will not be disclosed, but those are the 3 possibilities that is responsible for the difference in the storage temperature of these 2 RNA vaccines. RNA vaccines from Oxford AstraZeneca, the Sputnik, Johnson & Johnson, and Casino, we can see that the efficacy of these vaccines are not an RNA vaccine, but one unique thing with them is the storage temperature. The storage temperature is much more better. They can be stored at normal fridge temperature. And what is responsible for that, as we have said, is that DNA are much more stable than the RNA. So, and then coupled with the fact that we are using viral vector, the protective protein coat of the viral vector can also protect the DNA, which will confer more stability on the DNA. So they can be stored at a temperature of between 2 to 8 degrees centigrade. And then we have, recently we have Novas, which is another example of a vaccine using the conventional method, the protein subunit type. Novavax is based on that. And then we can see that its storage temperature is also similar. They can be stored at fridge temperature. And then we have the inactivated or killed SARS-CoV-2 vaccines from China, from Sinovac and Sinopharm. They also have similar storage temperature. And then, whereas efficacy and ease of distribution are the main focus in the design of this vaccine, the cost is another area that we have to look into. And then we can see that Moderna vaccine is the most expensive. And then we have AstraZeneca at a cost of $4 per dose is the cheapest, because AstraZeneca are saying they are going to sell their own vaccine at the cost price. So that is why it's relatively cheaper. Then we have the next that is cheap to this, the Sputnik V from China, and then the recent one, Johnson & Johnson. They are all at a relatively cheaper cost of $10. So those are examples of the different type of vaccines that we have. So what do we know and what are the unknowns of this vaccine? Presently, all the vaccines have demonstrated that they can generate robust immunity than natural infection, because we need vaccine. And just as I have said, vaccines are made, the principle behind it is to mimic infection in such a way that we can trigger immune response. And most of these vaccines have demonstrated that they can produce a more robust immunity than natural infection, which is very important for us. And then most of these vaccines have also been able to demonstrate that they can protect against symptomatic infection. But what we do not yet know is that whether they are able to provide efficacy in preventing against all infection, both symptomatic and asymptomatic infection, which is important for us if we have to achieve herd immunity. And then what is also not known is the duration of efficacy of the efficacy is the immunity. Is the immunity going to be lifelong, as we have with some vaccine like measles vaccine? once it is given, it is a lifelong immunity, or is the immunity going to wane over time in such a way that you need to give booster dose, probably more than 2 doses? So it is still not yet clear. So from what we have seen so far, can vaccine offer us an exit plan from this COVID-19? My answer is yes, it is possible, especially with the development of the RNA vaccine that we have seen. RNA vaccine from Pfizer and Moderna that emerged as a result of these have demonstrated a high efficacy of over 90%. So which is very encouraging, and it is showing us that it is possible to achieve herd immunity. Because for us to achieve herd immunity, we need a vaccine with efficacy of between 80 to 90%, and we will need to vaccinate approximately up to 70% of the population for us to be able to achieve this herd immunity. So it is possible for us to COVID-19 vaccine can provide an exit plan for us to exist from this COVID-19. So the RNA vaccine is a good development for us. It is likely to turn the tide in this pandemic, and it can unlock a new area of vaccine that can be used in a variety of viruses, like we have with the HIV, the Zika virus, and the RSV viruses. So the development, the emergence of this RNA vaccine is a revolutionary technology that is likely to expand in the future. And for us as a country, this is a time for us to invest in this technology so that we can also reap the benefits that is accrued to it. Thank you very much for your time.
Thank you very much. Thank you very much, Dr. Saad Abdullahi, for this very explanatory presentation. I believe that we have all learned so much from today's presentation. He has talked about the different types of vaccines and the vectors, and he has made us to be abreast with recent developments in the COVID-19 front. Before I go further, I want to welcome the chairman, Pharmaceutical Society of Nigeria, Kwara State, of the person of Pharmacist Adejumo Otelaja. Thank you for joining us today. I want to also welcome the chairman of ACPN chairman of Pharmacist Roland Tomiwa. Thank you for joining us. I want to specially recognize the HOUs, the head of units of University of Ilorin Teaching Hospital, the deputy directors. Thank you for joining us this morning. Every staff of University of Ilorin Teaching Hospital, our health professionals from different areas of practice here in Nigeria and outside the country, we are very happy to have you today. This is the time for our questions. I am sure we have a lot of questions today for the speaker. The floor is open. Please indicate by raising of your hand so that you'll be given the floor to ask your questions concerning comparisons of different COVID-19 vaccines. Please, you can unmute yourself. Dr. Faith, we can see your hand raised. You have the floor, man.
Good morning, everyone. I hope you can hear me.
Yes, ma'am, we can hear you.
Yes.
Okay. Thank you very much, the doctor who gave the presentation. Unfortunately, I couldn't because of work. I just wanted to ask, he was trying to tell us about the storage, the different storage of the, I think the Moderna and Pfizer vaccines. So I wanted to know the reasons he gave as to why we have the different storage ways for those two vaccines. I was trying to get that, but something took my attention. I just wanted the explanation again. Thank you.
Okay.
All right, thank you very
Thank
much,
you very
Dr.
much.
Faith.
Yes.
Yes,
Can
sir,
I
you can
respond to
continue,
that?
sir. Yes, sir, you can.
Okay. What we are saying is that the main reason or what is responsible for the difference in the storage temperature for the two mRNA vaccines from Pfizer and Moderna is probably due to differences in the design and modification of these two vaccines. It is possible for you to modify your RNA in such a way that you can improve the stability. One of that is by adding stabilizers and removing water from from the RNA vaccine after manufacture in such a way that it will increase the stability. When you add stabilizer and you remove water molecule from it, it will be much more stable. But this approach is also going to be to demand extra effort. Then we can also use chemistry. We can modify the 4 nitrogenous bases, for example, by adding a methyl group to any of the 4 nitrogenous bases. This can also modify the RNA vaccine. And then in the design of the vaccine itself, when you are designing the RNA, the genetic instruction, the part of the gene that you want to give that will trigger that immune response. The nucleotide, if you look at the nucleotide sequence of the mRNA of Pfizer, it is much more longer. It is about 4,200 and something long, whereas that of Moderna is about 3,819 nucleotide long. So the difference and the makeup of that 2 nucleotide sequence can also be responsible for the variation, maybe slight variation in the temperature or in the stability of the vaccine. So those are the 2 main areas. So it is possible Moderna use one or any of the 3 approach to improve the storage temperature of their vaccines. I hope that is clear.
Thank you very much, sir. Please, are there any more questions? If you have questions, this is the time to ask your questions. Please raise your hand and the floor will be open to you. Yeah. Okay, Dr. Bello, you are raising your hand, please. The floor is yours.
Thank you, Dr. Abdullahi, for that brief one. My question is on the vaccine development. You mentioned in your lecture that they are in the phase 3 of the clinical trials.
That they are
in the phase 3. I mean, those vaccines you listed are in the phase 3 of clinical trials. Are we?
Yes, most of them are in the phase 3.
Indicating that they are still under development and that the safety and efficacy are not yet 100% sure. Can you talk more on that? Any ethical concerns? Yeah.
Yes, we all know that actually phase 3 is the stage at which approval can be given for some of these vaccines. But we know we are in a pandemic situation. Most of these vaccines, they've not published peer-reviewed paper on the phase 3 trials that they've carried out. So it will take a long time for these vaccines to be approved because they have to look at all the published peer-reviewed journal for all the studies that have been done. These have not yet been done. What we have seen from the FDA is to grant emergency use authorization, which is different from outright approval.
Okay.
So that is to show you that they are still in the phase 3 development. A lot of development are still going on, and we can still have modification or perfection of some of these vaccines.
Yes. Thank you so much for that wonderful answers. I'm satisfied. Thank you.
Yes.
Thank you so much, Dr. Saad. I would like to ask a question. I would like to know which of the vaccines you would recommend for use in Nigeria, especially considering the stability. You talked about the RNA vaccines and the DNA vaccines being more stable compared to the RNA vaccines. But when I looked at your chart, you talked about the RNA vaccines being more efficacious. You said over 90% efficacy compared to the DNA vaccines. So which of them would you recommend for use in Nigeria?
Yeah, thank you very much. For us, for developing country like us in Nigeria, and taking into consideration the stringent storage requirement that is required for the RNA vaccine, there is no doubt that the RNA vaccine is much more potent, as we have seen, because this genetic approach is a more of like a targeted approach. It is not surprising that the RNA vaccine is much more efficacious because it's more specific, it's more targeted. So the efficacy is there, but for us in this part of the world, we don't have the logistic to store these vaccines, the challenges that will come with it. We don't have the facility on ground. So for us, it may be much more better for us to look at this vector vaccine, the DNA vaccine, especially the one from Russia, which has higher efficacy up to 90% because they have used 2 adenoviruses to improve the potency of their vaccine. And then it's also much more stable. So it is better for us to look at that aspect of the vaccine. The RNA vaccine, assuming we have a domestic manufacturing capacity for vaccine in Nigeria, it is also possible for us to make use of this RNA vaccine. It is possible with some countries like Australia that has this capacity. What they have been able to do with Pfizer or Moderna is to get license for the vaccine to be manufactured in their own country, so that you can bypass all these rigors of logistics, support of transporting, and all these challenges. So, but for us in this country, we don't have the manufacturing capacity for vaccine, and we cannot get license from all these first-class manufacturing countries like Pfizer and Moderna. So it may be much more better for us to look at the Sputnik vaccine from Russia, or the Oxford AstraZeneca vaccine that is also relatively cheap. Those two are relatively cheap and they are stable.
Oh, thank you so much, sir. Thank you. That was very explanatory. Pharmacist Ibrahim, you're raising your hand, sir.
Thank you very
much.
Thank you very much, the coordinator. And I also want to thank the speaker for a very well presented paper on vaccines. As healthcare providers, we have a duty and responsibility to convince our people on the safety of the vaccines. And this has been well demonstrated from the presentation that they are safe, and the types of vaccine and capacity to store the vaccines. My own question has to do with the public enlightenment. At one point, while trying to enlighten the public, a question arose, from members of the public that there are some of the vaccines that have been specifically prepared or labeled for use in Nigeria or used in developing country. And I then find it a bit difficult to answer such questions. I wouldn't know whether the speaker may have an idea on how to respond to such questions, but they are very important to be able to carry everybody along on the safety of these vaccines. We are being made to believe that there are certain vaccines that are actually designated for use in developing countries. Though I've not seen such, but then these are questions from the public, and we as healthcare providers should be able to answer these questions. Thank you very much.
Oh, thank you very much, sir. If I get you right, the issue of some vaccine being designated or labeled for use in a particular country, as we all know, may be attributable to the fact that some of these vaccines are maybe manufactured specifically or with fund from a certain organization, non-profit organization, and so on. And so because of that, there may be restriction that, okay, this vaccine is made particularly for this part of the country and so on, and it has nothing whatsoever to do with the safety of the vaccine. I don't know, there may be much more better explanation, but we have examples of that, okay, this particular vaccine is produced with donation from certain organization, and then there may be restriction to the circulation of that vaccine that, okay, it's only meant for this part of the world and so on.
All right, thank you very much, sir.
Hello.
Yes, sir, can hear
Okay.
you. Thank you so much, sir, for that explanation.
Yes.
Do we have any more questions? Please, more questions before we leave the questions and answers. Do we have any other questions? Please indicate by raise of your hand. All right. In the absence of further questions, I want to particularly appreciate Dr. Saad Abdullahi. Thank you for expatiating and expounding on today's topic. You did a very wonderful job on it. Thank you for keeping us abreast with COVID-19 vaccines available right now in the world. We are very, very much appreciative. The Department of University of Ilorin Teaching Hospital, Department of Pharmacy, University of Ilorin Teaching Hospital appreciates you. I want to recognize some persons here present with us. I would like to recognize Professor Ibrahim Oriagba. Yes, I would like to thank you for joining us today. Thank you, Professor Olufunke Sodipo. I would like to thank Pharm Bola Mustafa. Thank you for joining us on today's presentation. Pharm Nuruddin Alabi, fellow PSN, thank you for being here today. We are really happy to have every one of us here. Thanks for joining on today's presentation. I saw the Dean of Pharmaceutical Sciences, University of Ilorin. Thank you for being here today. We are grateful. I want to thank Pharm Dr. Bello of University of Ilorin Teaching Hospital, Drug Information Unit. Thank you for putting this together. As we round off today's presentation, we'll have one like this same time next week. Please tune in. Thank you all for being here, distinguished colleagues from different areas of practice, all other health workers. Thank you for joining us. We are grateful to have you. This is the end of today's presentation. I will be handing over to Pharmacist Bello to continue from here. I am Pharm Dr. Usifo Ngozi, the moderator for today's presentation. Thank you very much.
Thank you.
Pharm Bello, please.
Thank you, Dr. Ngozi, for that one. A very good one. Thank you so much. We appreciate you. We want to ask us to drop our email address, and we promise that the video recording and the slides of this presentation will be shared with us by God's grace. We thank you for joining us. We'll say bye for now. Thank you all.